Hey there, science fans. Today we're diving into the exciting world of biotechnology and uncovering the secrets of life itself. It all starts with the central dogma of molecular biology, a fancy way of saying how information flows in living things.
Imagine DNA as the blueprint of life. It's a long, winding molecule tucked away inside our cells, carrying all the instructions to build and run our bodies. Think of it like a massive cookbook with recipes for everything from your hair color to your digestive enzymes.
But DNA doesn't do the cooking itself. That's where RNA comes in. RNA is like a messenger carrying copies of those DNA recipes to the ribosomes, the tiny chefs of the cell.
Ribosomes read those recipes and use another ingredient, amino acids, to build proteins. And what are proteins? They're the workhorses of the cell, doing everything from digesting your food to fighting off infections.
So there you have it. DNA to RNA to protein. That's the central dogma.
Let's take a closer look at that first step, from DNA to RNA. This process is called transcription, and it's all about making copies of those important DNA recipes. Picture a scribe carefully copying down instructions from a giant scroll.
Inside the cell nucleus, an enzyme called RNA polymerase acts as our scribe. It binds to a specific region of the DNA molecule and starts reading the genetic code. As it reads, it builds a single-stranded RNA molecule that's a mirror image of the DNA sequence.
This newly made RNA molecule is called messenger RNA, or mRNA for short. It's like a single recipe card, carrying the instructions for one specific protein. Once the mRNA is complete, it detaches from the DNA and heads out of the nucleus, ready to find a ribosome. Now it's time for translation, the process of turning that mRNA recipe into a protein. Think of the ribosome as a bustling kitchen with the mRNA as the recipe card.
The ribosome reads the mRNA code, 3 letters at a time. Each 3-letter code, called a codon, corresponds to a specific amino acid. Like a skilled chef gathering ingredients, the ribosome recruits transfer RNA, tRNA molecules, each carrying the correct amino acid.
As the ribosome moves along the mRNA, it links the amino acids together in a chain, following the recipe precisely. This chain of amino acids folds into a specific 3-dimensional shape, determined by the sequence itself. This folded structure is what makes a protein functional.
And just like that, we've gone from DNA to RNA to a fully formed protein.
Scientists have figured out how to harness the power of DNA and RNA to do some incredible things, like diagnose diseases, develop new medicines, and even solve crimes. One of their most important tools is called PCR, which stands for polymerase chain reaction.
Think of PCR as a DNA photocopier. It allows scientists to make millions of copies of a specific DNA segment in just a few hours. How does it work? PCR relies on a special enzyme called DNA polymerase, which naturally copies DNA in our cells.
The process has 3 main steps: denaturation, annealing, and extension. In denaturation, the DNA sample is heated to separate the 2 strands of the double helix. Then, during annealing, short DNA primers bind to the target DNA sequence. Finally, in the extension step, DNA polymerase uses the primers as starting points to copy the target sequence.
This cycle of heating, cooling, and copying is repeated many times, resulting in an exponential increase in the number of DNA copies. PCR is like a molecular magnifying glass, allowing scientists to study even tiny amounts of DNA.
It's used in everything from medical diagnostics to forensic investigations, making it one of the most important tools in the biotech toolbox.